please.
Thank you very much for the introduction, and good morning, everybody. First of all, I would like to thank the organizers to bringing me here. I enjoyed the meeting so far very much, and I hope that I can share with you the excitement we had regarding the work that was done together with Virginie in Montpellier. Virginie is working in a department of agriculture, the INRA, and therefore she's supposed to do stuff that is related to food science. Okay, I'm not into food science except that I like to eat, and but Virginie is a good scientist, and therefore she said, okay, but we can do still something together. And so that was the origin of this work. It's about the rigidity of plant cell. This is what we are saying if we want to get the money, and so that Virginie is justifying her existence. As you will see, we will boil down this to a rather abstract problem that is perhaps much more interesting, but at least much more simple. So this is a cartoon of a plant cell. The real stuff, of course, looks much more complicated. And so what you have is some all sorts of structural elements that are responsible for the stiffness or the mechanical rigidity of the cell. But among other things, you have what is called pectin. So pectin is the stuff that if you cook fruits, that makes this gelation. So pectin is, in fact, a polymer. And here there is, on the left-hand side, pectin. It's a longish polymer. There are various sorts of pectins. At the end, we don't care about it. And so nature makes that pectin is transformed via what the biologists call an enzyme. And we call it a catalytic. And therefore, so basically, it's this rather complicated object that comes and attaches to one of these monomers here, does something to this monomer, and rends this monomer attractive, okay? So first you have here a long chain of this pectin that is basically neutral. There are only van der Waals interactions. Then comes this PME, the pectin methylesterase, and transforms locally one of these groups, and then this group attack each other. And since you have then now a chemical attraction, so this polymer soup starts to interact strongly with each other, and it forms a gel. And this gel formation is responsible in part for the mechanical stability of the cell. Okay? And so that's what we want to study, what is actually happening, how is this gel formation. So as you see, this is complicated, this is even more complicated, and I was a simple-minded physicist, therefore I do this. So this here is now our pectin, and the Pac-Man here is the colloid, which basically comes and attacks one of these monomers, transforms a blue monomer that was basically repulsive into a red monomer that is attractive, and then it goes away and attacks another one, etc. And so with time, therefore, you get more and more of the red monomers, since the monomers attack each other, and the polymers that at the beginning had only steric interactions now start to come together and form chemical bonds that are strong, and therefore this is the formation of the gel. And that's the system that we're going to study. So this is now simple enough that even I can try to do something. And to study this, we are using computer simulation. For the polymers, we are just taking a chain of soft spheres. So soft spheres is nothing else than a purely repulsive potential r to the minus 12. 12 is not a magic number, it's just convenient. And in the following, we will call these particles that are repulsive, so the blue ones, aparticles, okay? And then in order that they stay together, in order to form the chain, you include also for the nearest neighbor a so-called potential, which is basically a glorified harmonic spring between the particles. Okay. So this interaction here, the steric interaction plus this harmonic spring-like potential gives rise to the polymer chain. The colloids, these are just soft spheres that are twice as large as one of these monomers here. Why twice? Because this is a reasonable number. Again, there are various types of enzymes. bigger ones, smaller ones, etc. This was something that was convenient also from the simulation and not crazy from the point of the physics. And then once now you have that the enzyme attacks one or transforms one of these blue monomers, the monomer is transformed in a continuous manner into a particle that interact with each other via Lennard-Jones potential. So now you have a finite interaction, positive interaction. So that's the red particle, and I will call them in the following B particle. So at the beginning, you have a soup of polymers that are completely composed just of A particles, and with time, these A particles are transformed into B particles, and therefore the chain starts to interact. And so the length of the chain is 100. So this is decent size. It's not huge as in reality, but you are already beyond the entanglement length. That is to say, you are basically in the right rheological regime. 400 chains, and what you are doing is molecular dynamics to study what is happening with the system during the ongoing reaction. Okay. Yes. Okay, that is an important point. So basically, so you have two blue particles, no, a blue, excuse me, and a green particle. And so once you come closer to a certain distance, then the blue one is transformed into red, okay? And this is, thank you for the question, because it's important that in fact there is at the beginning some sort of a repulsion, and then you have to overcome this repulsion, and then if you have come close enough, you can flip it over. I'm sorry. Yep. It still remains red. Yes, then once it's red, it's red forever. Okay? No, it's a catalyst. So it's you know, it goes on and does the damage elsewhere. Okay. So what do we expect? So basically, as we said, so we start out with many A particles that are forming polymer chains, and then with time we get more and more attraction between the polymers. Now, imagine here a standard phase diagram with temperature and the density of the B particle. So at time 0, you have 0 B particle because you have not started the reaction yet, and therefore the density is very low. So you have, let's say, at a certain temperature, T2, and you are down here. And now imagine that you get more and more of the B particles, and so in principle, you can expect that there is a phase coexistence between a one-phase region and a two-phase region down here with a critical point here, okay? Because our simulations are done below this critical point, it's really interesting. And so the system marches from the left to the right because it gets more and more B particles, that is to say, the interaction is increasing more and more. And therefore, at the beginning, you have a few B particles that are in a sea of the A particles. And then once you hit this coexistence line, the system in principle becomes unstable. It should phase separate. But usually there is a metastability. You go on, hit the spinodal, and then it really becomes unstable. And then it should really start to phase separation. So what we expect is some sort of phase separation dynamics. But here it's not because you have done a quench in temperature and weighted. That's usually what you do in phase separation. But here it's because we are changing the interaction between the particles. And therefore, we are at constant pressure, temperature, and we are just marching to the right. So in the following, we will see the influence of the temperature on this reaction is set. Now, once you are at low temperature, then you can expect that okay, you are transforming, but these here are polymer chains. These are not simple liquid particles, and therefore, at low temperature, yes, you have transformed, but due to this polymer connectivity, the system will have a hard time to phase separate. And therefore, probably here you will fall out of equilibrium and therefore form a gel. And so this is basically the gel formation process that we are going to discuss today. Okay, now in the following I will give you some key quantities in order that you understand what the system is doing. and then before we put all together into a nice uniform picture. So one key quantity is certainly the concentration of the B particle, of the attractive B particle. And so this here is time, and here is this fraction for various temperatures. And so what you see is that at the beginning, you just have a linear dependence. So this is t to the 1. This implies that basically the green particle finds at each time a new A particle that can be transformed. So at each time unit, some certain damage can be done, and therefore the concentration of the B particle is just increasing linearly in time. But then at long times, the colloids or the enzymes do not find these A particles anymore because they have been eaten up. And therefore it has to search a little bit longer, and therefore there is some sort of slowing down before ultimately all the A particle has been eaten up, and therefore the blue phase has been transformed into the red phase. Okay, so, but the message is, so it's a linear dependence at short times, and basically this linear dependence is seen at all temperatures that we see. And the second message that you learn from here is that you decrease temperature by a factor of 3, and you get a huge slowing down in the dynamics. That's nothing that surprising, because it is an activated process, as you will see. Okay. Now, this was at a given concentration of the colloids. Now, since we saw that the colloids are wandering around, they making damage, they go on, etc., what happens if you increase the concentration of colloids? So at the beginning, if you have a very low concentration, basically the colloids, they do not see each other. Each one does its own damage here and here, and therefore if you have twice as many colloids, the reaction dynamics is twice as fast. And therefore, the relevant quantities to look at is probably times the concentration of the colloids. So it's not the number of colloids, but the concentration of colloids. And you can hope that a sufficiently low concentration, basically, if you trace the previous curves as a function of this rescaled time here, you will find a master curve, and this is indeed what you find. So these here, the colors are different temperatures, and the different symbols are different concentrations. So let's take for the lowest temperature to start with. So if you go from 0.5% to 1% to 5%, basically you see at the beginning they fall on top of each other, and then if you have a higher concentration, then you fall off this master curve. That is to say, this answer is no longer good. But as long as you stay in the low concentration regime, this is really the relevant time scale. And that's what you're going to do in the following. We will stick with low concentration, so that basically we have independent walkouts. Okay. Now, what about the influence of density? So here, the different curve colors correspond to different densities. Let's start with a low density, the green one, and here for different temperatures. So you see that if you go, let's say, from green to blue to red, green, blue, red. So if you increase the density, then things accelerate. And it makes sense because the random walker or the colloid has to walk less far in order to find something to transform. This is interesting, this is trivial. What we found, and which we have not really understood, is that the dependence is basically exponential in rho. And so we would have rather expected some sort of a power law, but it's a quite good exponential, but frankly speaking, we have not understood where this comes from. So we just leave it as it is. And this behavior here is basically observed for all temperatures, so temperature, in a way, is not a relevant quantity. Okay. Now, let's become now a little bit more quantitative. So these are the curves that I showed you, and since they are all linear dependencies at short, small concentrations, what you can do is to shift them vertically and try to produce a master curve, and this is what is done here, and you see that they collapse on the master curve. And so you have here the shift factor, R of t, that depends now on the temperature, that is used basically to obtain this master curve. Now, the shift factor tells you by, apart from this trivial dependency here, how much does temperature affect the transformation? So a constant FB, that is to say constant concentration of the B particle, basically tells you now the system from the thermodynamic point of view has the same number of attraction, and therefore this is some sort of a driving force, or the R of T characterizes, gives the time scale to reach the same thermodynamic driving force, because the concentration of the P particle is nothing else than the driving force. Now, how does this R of T look like? It's just a simple exponential, that's what we find. And with this exponent here, around 3 as an activation energy. And this activation energy matches very well the one that, and this is why I was happy that Google was asking the question, this activation, this barrier here, is nothing else that the colloid has to overcome in order to come close to the blue particle. And so it basically has to run up. Sometimes, or more often than not, it fails, especially at low temperature. But if it comes, then it can transform, and this activation energy is found here now in the temperature dependence of the reaction. So very simple and very clear how this works. Now, how do this system look like? So this here is the system at the beginning. So the big blue particles, these are the colloids, and the small blue particles, these are the A particles. That is, say here, this is the polymeric system, homopolymers. There is no B particle inside. So no attraction, these are purely repulsive particles. And then you start to move on in time, and then you get still the blue particle, the gray as the A particle is still around, but now you see that you have here a few red particles. These are the attractive B particles. I have to show you this. Playing, yes. And so basically you see that you have here this disordered structure, which sometimes starts to have a little bit of holes. That is to say, now it's no longer homogeneous. Okay? And also what you see is that the colloids, here they were dispersed into the system in a homogeneous way, whereas here they start to cluster a little bit. And this is a purely entropic effect. The system is contracting because of the attraction, and therefore it forms holes, and the colloids are taking entropic advantage of these holes, and they go inside. Therefore, you have this sort of microphase separation, not complete, but tendency that the colloids go and fill up the holes. Now, this is what happens if you do the reaction at low temperature. If you do the reaction at high temperature, then it's way more boring, because then you get just a simple phase separation. So all the blue particles here, the light blue particles are transformed into red. The system completely phase separates, and the enzymes are expelled from the system. The reaction basically stops, the system is dead. So this is boring. We want to study this one. True, yeah. But I mean, this is our finite simulation cell. If you would have a bigger system, then and you see it's still hanging on. So these are a few polymer strands that do not want to decide whether it wants to be on this part or on this part. But with long enough time, basically, you would expect one big blob at the center surrounded by a halo of colloids. Okay. Now, no, it's a one-way street. Now, so this here is in a way the final snapshot. Let's quickly look at the dynamics. So this is at the intermediate temperature. So you start out with a homogeneous soup, and with time you will see red particles that appear. And so this here is then the ongoing reaction. And so you see that basically there, that's where the simulation stopped. And so basically you see now you have some sort of a very heterogeneous structure where the red particles, these are attractive, and you still have some gray particles non-attractive. But you see this, okay, this looks like a gel, why not? Now you do the same thing at low temperature. Now please, I should point this out, So if it looks like a gel, it means this looks like a fractal. People have seen this before. Why should I bother you with this? It's because I want to show you the contrast to low temperature. So this is what happens at low temperature. And so at the beginning, nothing exciting. It seems to be almost the same. So you have to need a little bit more patience. So now the red particles, the attractive particles start to form. And so, but if you compare these two things, then here you see this is some sort of a fractal, but this here, this looks much more granular. And so on the next slide, I will show you just the red part of it. That is to say, only where are the attractive B particles. And this is this. And so you see that, in fact, this is no longer a fractal structure. This is a cluster phase. And so somehow the system managed to set up or organize itself in a way that is very organized in that it finds this micellar structure. Okay? And so we have this mesophase. Inside that is watered gel. I mean, here, everything what is white is filled with the other particles. So it's not that you have empty space, but if you would make a scattering experiments, you would see a strong contrast between the red and the empty. And so in the following, it will come. I'm here to answer all your questions. Okay, before, let's see, in order to characterize the lifetime, we want to see, well, what is actually there? So this here is the cartoon of a snapshot of the structure that we have. And so what we found by looking at these movies and at snapshots, etc., that it seemed that whatever the temperature, if you are at intermediate or low temperature, and you let run the evolution of the system, then we saw that there was always this cluster phase. And so we were thinking, well, is this really universal? In the sense that for all systems, there is this evolution of this cluster phase. And therefore, what we defined is a new time scale. Sorry about this. So you know the TB, this was the time scale that was used to look at the concentration of the B particle. So basically, this was related to the thermodynamic driving force, okay? Because it tells you how much attraction that you have. But once you have an out of equilibrium system, and you have a thermodynamic driving force, the system does not, or you do not really know how the system will react to this driving force. So it might react quickly or not. And so this additional time scale, multiplicative factor here, tells you whether the system reacts quickly, so if S is small, or whether it reacts in a sluggish manner. And so this, in a way, is some sort of dynamic susceptibility. You have driven the system out of equilibrium, and the system will react to it, and therefore you have picked up this time scale. And so the way we define this time scale is to look at structure. Therefore, this is basically an isostructure time scale. We are looking or defining the S of t so that if the Ts is the same, the structure of the system is the same. And it's not trivial at all that such a time scale exists. Okay? And so we checked it. And so at time equals 0, of course, it exists because nothing has happened. Then here you have started a little bit, and then you see that you have here, so excuse me, this is the static structure factor for the B part of the red dots. And so you see that you pick up here a structure. The nearest neighbor, excuse me, I should have said this, the units here are reduced units. The nearest neighbor peak gives rise to a peak in the static structure factor at around 2 pi. That is to say, this peak here is the nearest neighbor distance. So what we see, however, is also a peak at much smaller wave vector, and this peak is related to these clusters, okay, which have a much larger distance from each other. Well, much larger, about 6 or 7 particle diameter. And so what you see, yes, it's the distance. It's the distance because you see at the beginning it's still very fuzzy, and then it becomes a bit sharper, so it's really the distance. It's the distance that does not change, but the shape of the cluster changes. And so what you see is that with ongoing time, you always find a combination, that is to say one of these factors, so that you can superimpose the static structure factor. That is to say, although the dynamics of the system in real time is very different if you change the temperature, you always can rescale or find a rescale time so that the dynamics in this reduced time is the same. And so this either structural time really exists. Okay, now come to Chandan's question, how long do these structures really exist? So what we are looking here is, excuse me that I want to go back, is at the intensity of this first peak, because this is a good measure of the internal clock of the system. So how high, what is the height of this peak? And so we plot this height as a function of this reduced time here, and by construction we find that for, let's say, at this density and the different temperature, we can get a superposition. And so what you see is that at the beginning there is a rapid increase, and it's basically exponential in time. Then it flattens off, and that is to say this cluster phase is relatively stable. Before then, let's say at high temperature, the red line here, the system really phase separates, and then the cluster phase is destroyed. But if you are at low temperature, the shoulder here went on until we ran out of patience or out of money paid by the computer. So since there is this master curve here, I would answer to Chandan, if you go to sufficiently low temperature, this plateau will live forever in the day. So it's really a stable structure. Okay? And what you also see is that if the density is large, then this reaction happens quicker, and therefore, this is shown in this graph here, that you gain about 2 orders of magnitude by changing the density by a factor of 3. Okay. But again, we do not understand where this density dependence, why this density dependence really comes from in a quantitative. Okay. Now, what this scaling factor here, that we have here, the S of t has a certain temperature dependence. Here, sorry. And we find again that it's an Arrhenius law. And the activation energy is around 1. It depends a little bit on density, one has to say. But at high densities here, these points here, it's a little bit higher than 1. And 1 is basically nothing else than the interaction between two attractive particles. That is to say, the driving force. So you have now a certain concentration of attractive particles, but they are not in an equilibrium configuration, and so they want to rearrange themselves to reach a better thermodynamic state. And the time scale to do this is basically just governed by the depth of the well, and therefore, basically, in a semi-quantitative way, you really understand also this part of the dynamics. Now, to wrap the thing more or less up, I'm changing, in a way, a little bit the viewpoint, and we are going back to, in order to understand what is really going on in the system, and we go back to rather old work, first by Fredrickson and Milnor, and then by Song and Jazirat, that looked at random block copolymers. So what is this? So this is something that really exists in nature, or at least in the chemical industry. So basically you have 2 types of monomers that you can call A and B. And so you start your polymerization process with an A monomer, and then you have a certain probability, excuse me, you have A polymer with a certain concentration, Fa, and you have the B polymer with a concentration 1 minus Fa, Pb, excuse me. Then you start, let's say, with an A monomer, and then you have a certain probability to attach to this A particle another A particle. This probability is given by PAA, or you have a probability to attach a B particle, so PAB. And if you then have a B particle, you have a probability P BA or a probability BB to attach the next particle. Okay. So once you have therefore the concentration of the A particle and these two probabilities, you have defined your system, your polymer, and what these people showed by means of field theoretical calculations, and Sung and Jesse Raj, they did some more sophisticated liquid state theory. They showed that this polymer mixture can be homogeneous or demixed depending on these parameters and the temperature. And so, so we have here therefore a random chain of A and B monomers that have a certain statistics, and this smelled similar to the stuff that we have. Therefore, we wanted to check, is our system that is not produced at all in this manner here, but can it be described by this model of a random block copolymer? And it works, else I have taught you all about this. So basically from the simulation, so you take a snapshot of your system, therefore you know how many A particles you still have. You can look at the PAA and the PBB, so these are probability that you just measure from the statistics of your polymers. And now you can ask, if you take any one polymer and I look, what is the length of a segment, let's say, of pure A particles or of pure B particles? And you look at the probability to find the segment of a certain length for the A or the B, then you will find certain distributions. And so the field symbols are the results from our simulations, and the open symbols are from this model here. And so you see, so there is no fit parameter because we have measured these things here, and the rest is just mathematics. And so you see that with a very good approximation of accuracy, the open and filled symbols agree with each other for the A length distribution and the B length distribution. That is to say, what we have realized is, without knowing it, to say the truth, is a random co-block. Okay? And therefore now we can use the results that these people got and compare them with our phase separation. And for this, I quickly want to show you how do the parameters that are inside this model depend on temperature and on time. And you see that, let's say this is this probability PAA, and you see for the different temperatures, they all fall on top of a master curve. Why it looks now exactly like this, I don't want to go into the details. The black one is the probability of the B, the green one PBB. So these are basically the relevant parameters. Lambda is, for historical reasons by the theoretician proposed, this combination here, this is the blue curve here. So the message of this curve here is that that basically all this temperature here give rise to the exactly, or to a good master curve. That is to say, indeed, this is other evidence that indeed the structure of the polymer can be described on this time scale. Not the dynamics, this is the time scale Ts, but the structure is given by the time scale Tb. And now, Sung and Jesse Raj, they did calculate the phase diagram. So this here is basically a parameter that tells you something about the statistics within the chain. And this here is the chi parameter that we have already encountered yesterday. So it tells you whether the two polymers hate each other or love each other. So if they love each other, they stay homogeneous. Whereas if they hate each other, they phase separate. Okay? So you have here a phase transition between a homogeneous and a heterogeneous or inhomogeneous phase. And so depending on the concentration of the A particle, of the blue particle, you can, the authors, the theoretician calculations predicted two different types of theory that looks within 10 or 20% basically the same. So the interesting part is really this rising here and then it comes down. Now the colored symbols here, these are our simulations. And so what we find is that, again, in this quantity here, we fall onto a master curve for all densities investigated. So this is nice. So it really shows that this transformation dynamics is very universal if expressed in these parameters here. And you're crossing these points here. The blue line, the theoretical line around this here. And so if you look then in the real, now real data here, now on data, at what time it happens, you cross it here basically when you are, let's say for the 0.6 at this point here. Is to say, here you're growing, nucleating something, and then this plateau here, you have entered the inhomogeneous phase. And so this here is therefore nothing else that spinodal instability that was predicted long time ago by this field theoretical calculation. And that, to my knowledge, has not been really seen in computer simulations because of bad choice of parameters, whatever. But here we see, without, we didn't look for it, we have here now an example of such a random block copolymer that really shows this phase separation. The theory was unfortunately not able to predict into what phase it separates. They said it's homogeneous here and inhomogeneous here, but then figuring out what is the new phase, this is very complicated. So they were not able to tell it. And now our contribution in this direction is that we know that it's really this cluster phase that is very regularly spaced. Okay, and with this, I'm almost up, and therefore I want to conclude. So basically, what we have proposed here, or looked at, is at a rather unusual way to form a gel. Usually, you form a gel by taking a simple liquid and you quench it to low temperature, or by throwing in increasing or the density or something like this. Here we have a catalytic reaction, that is to say, we are changing effectively the interaction between the constituent particles. And so, since it is slow dynamics, that we really get a compact gel, but that has an internal structure. It's to say, this mesophase of the clusters. Since we have seen that the dynamics of transformation can be understood semi-quantitatively, because we have understood all the ingredients, the barrier heights, etc. And therefore, from this point of view, this work can guide a little bit also the experiments to get into the good temperature and time regimes if you really want to generate such mesophases. And the nice surprise is that the chemical structure of the system that we looked at turns out to be very close to this random block copolymer, and that we can use the theoretical prediction on the phase existence of the stability limit of the homogeneous phase to understand why our system is really phase separated. So this is very recent work, and there are of course many open questions. So what we did was a Newtonian dynamics. In real life, probably you will want to take an overdamped dynamics because you have viscosity of the solvent, etc. What is the role of the size of the colloids? Because these enzymes, they come in all shapes. They come small, bigger ones. What we are dreaming about, is it possible, for example, that you take relatively big colloids and the formation of the network due to the catalytic reaction will trap these colloids and therefore poison and stop this reaction. And therefore, you can basically trap nanoparticles inside this matrix. And at the end, we should also reconnect with the biological system and see whether experiments can be done to see this phenomenology. Okay, with this, time is up, and thank you very much for your attention.
Okay. Okay. So, you know, this cluster phase you talked about somewhere in the middle of the talk is reminiscent of also what people have seen and studied in systems with short-range attractions and longer-range repulsions. So one question is this a related phenomenon that's interpretable in the same terms?
I think so.
And Second question, I lost a little bit of the biology in the beginning, and therefore the motivation for you to do the modeling exactly in this way. So could you not equally well have used a potential of that kind for what you wanted to study?
Well, you mean the potential for the polymers, or for the overcome the barriers? Or?
Overcoming the barrier. So, I mean, the way you explained it is something that would make me immediately think of a potential that has a repulsive barrier and an attractive well. So why not use that is the question.
Okay, so this is the real stuff. The reality is that the enzyme comes to the pectin, and it may react or not and it depends on the orientation of the enzymes. I don't want to simulate this. It's a prefactor that costs a lot of computer. So out. And so there is this here is not a sphere, it's an odd-shaped object, but from the point of view of physics, it's irrelevant. Because first of all, we want to understand what is the phenomenon that the experimentalists, they look at it and they do rheology as a function of time, and then they see something. And then they change certain ingredients and they see something else, and there is not that much understanding. So we wanted to be very modest regarding the model, but at least understand something. Now, yes, we have to connect these two worlds, and now if you are not happy with soft sphere potential, but I think from the point of view of insight, this is the first step to do. Now, next step to follow.
Yeah, hi. So I just had a question, regarding your cluster phase. If you had, you have flexible polymers.
Yes.
But if you had semi-flexible polymers, let's say, then of course the clustering process might be very difficult.
Absolutely.
You can't bend over surface.
Very good. Yes.
And so therefore the question is that would this, from the biology point of view, is it a flexible polymer, or do you not know? For example?
Well, the Kuhn length is several of these blobs here. So it's semi-flex. It's not rigid. It's not actin or something. But it's really forming a gel. So I do not know whether you really can see it for a semi-flexible polymer. I think it would depend on the length scale. But for good reasons, I did not tell you why is the length scale 1 or 6? Because I don't know it, okay? So that there must be some mechanism that picks this length scale. And before I have not understood it, I've done the simulation of the semi-flexible thing, which is of course a good suggestion. I cannot say whether this cluster phase is it. I see no reason why it should not, because it's a question of length scale.
I was thinking if there is a simple way to understand this fraction of B particles as a function of time. So at short times, can I just think of like a set of A particles diffusing, and then these green particles come and convert them into B. And so I just want to look at the surviving fraction of A particles. I mean, so is there a simple way to understand this plots?
So basically you are talking about this part here. I do not know what it is. We didn't look carefully at it because here it's almost starting to phase separate, and therefore basically it's some sort of competition. The colloids are expelled into the voids, and then you are touching only the surface. What we think is more interesting is this part here at low temperature, and as you see, we have bad statistics and we are not really reaching the asymptotic range. And that's why unfortunately, I'm not really able to answer. It is an interesting point because it tells you something about survival of random walks, etc. But too difficult at the moment.
Yeah. So Walter, this has nothing to do with the simulations you're doing, but the first picture that he had, the cartoon that is there some understanding of the orientational order of these cellulose microfibrils? You didn't talk about that, but they look very similar to these other phases of DNA membrane.
No idea. You're talking about the beans here, the microfibroids. I do not know what, I'm not a biologist. Ask Virginie, and perhaps she knows, but I'm talking about the yellow stuff.
Right, I know. But
Nothing else. And there are plenty of people in the room that know much more about the system than I do. The people, I don't know.
But it converts, I mean, a physics problem related to that has to do with polymeric pneumatics. So that might be an interesting.
I think it's this here.
So I'm not my field at all, so just tell me if this is a ridiculous question. But your cluster phase, is it the case by construction that it's above a percolation threshold of some sort? I mean, I can imagine mechanical stability of such a structure would depend critically on whether it really was a percolating network or whether it was fractionating. And some of your snapshots to me looked like they were clearly above the percolation threshold formed a connected network.
Everything is.
And others looked like they might, you know, the red guys were.
I agree. Yes. So I think if you're in this regime here, everything is fully percolating, etc.
Yeah.
And out here, especially at higher temperatures, it's basically falling apart. There, it's really undergoing phase separation in a stupid way, and so it's mechanically unstable. But here, I think you can walk on it if you are not too heavy. If you are not too heavy, you can walk.
Yeah.
Okay, so